Melanism is a rare but mainly naturally occurring condition that has been observed in many species, from leopards and jaguars to snakes and birds. But what causes it? How does it, simple genetic change, affect an entire organism's appearance, and more importantly, its survival? Today, we're exploring the science behind melanism to uncover the answers. Hi, I'm Shriyan.
And I'm Anish, and we'll be guiding you into the true beauty of melanism. Have you ever seen a black panther stalking through the night? Its sleek black coat blends seamlessly with the darkness, making it almost invisible to prey. Or have you ever seen a dark furrowed squirrel running through the trees? And have you ever noticed why it stands out from its light-colored relatives? Well, these animals might be different from their species, but what if we told you that this dark coloration wasn't just a coincidence, but rather it was a result of a fascinating genetical mutation known as melanism? Before we go into melanism, we need to understand what a mutation is. So, a mutation is a change in the DNA sequence that can alter the function of a gene. DNA is made of 4 chemical bases: adenine, thymine, cytosine, and guanine. And in RNA, the thymine is replaced out with uracil.
These bases create instructions that determine every characteristic of a living organism, from its eye color to its ability to fight diseases. Mutations can occur in several ways, such as substitution, insertion, or deletion of nucleotide bases in the DNA sequence. These changes can be passed down, or can even happen randomly during DNA replication, or can even be triggered by environmental factors like UV radiation or chemical exposure. As you may know, some mutations are neutral, some are harmful, and some, like melanism, can actually be beneficial. An example of a neutral mutation is your eye color. Most of the time, your eye color will not harm you or benefit you.
Melanism is a mutation that affects pigmentation, making an organism darker than they usually are. This happens due to an increase in the production of melanin, the pigment that gives color to skin, hair, feathers, and scales. In many cases, melanism provides a survival advantage, such as better camouflage or even increased resistance to UV radiation.
But what exactly causes melanism at the genetic level? Scientists have discovered that one key player in this process is the MC1R gene, short for melanocortin-1 receptor. This gene acts like a switch, controlling the balance between 2 types of melanin: eumelanin, which produces black or brown pigmentation, and pheomelanin, which results in a red or yellow pigmentation.
In a typical animal, the MC1R gene turns on and off as needed, regulating melanin production. However, in melanistic animals, a mutation in this gene causes it to stay permanently on, flooding the body with eumelanin, leading it to characteristic dark pigmentation. But MC1R isn't the only gene involved. Another important gene is TYR, which codes for tyrosinase, an enzyme that plays a big role in melanin synthesis. Tyrosinase is responsible for converting the amino acid tyrosine into melanin. If this enzyme isn't working correctly, melanin production is disrupted. Mutations in TYR can lead to either an overproduction or a deficiency in pigment, influencing the animal's overall coloration. For example, while melanism is caused by an increase of melanin, another condition called albinism is caused by a lack of melanin. Both of these traits occur due to different mutations in the same pigmentation track. This shows how powerful genetics are in shaping physical characteristics of an organism.
So, now that we understand what melanism is and how mutations play a role in its development, let's take a closer look at how this genetic change actually works inside a cell. To do that, we need to explore 2 crucial processes: transcription and translation. But how does this mutation actually work? To understand that, we need to explore a fundamental process known as transcription, the first step in turning genetic instructions into actual proteins. Think of transcription as a way for cells to read their DNA without directly using the original copy. It all starts inside the nucleus of a eukaryotic cell, where DNA is safely stored. Since DNA itself can't leave the nucleus, the cell needs a way to send instructions out to it. This is where transcription comes in hand. An enzyme called helicase acts like a molecular zipper, unwinding the tightly coiled DNA double helix so the genetic code can be accessed. Once the strands are separated, another enzyme, RNA polymerase, moves in. This enzyme is the true workhorse of the transcription. It reads the DNA strand and builds a complementary strand of messenger RNA, or mRNA. DNA is made up of 4 bases: adenine, thymine, cytosine, and guanine. However, RNA is slightly different. It doesn't contain thymine. Instead, uracil takes thymine's place. So, as RNA polymerase constructs the mRNA strand, it follows a specific base pairing rule. A pairs with U, C pairs with G. Think of DNA as a recipe book containing every single instruction for making foods. But instead of having the entire book, you make a handwritten copy of just one recipe. That copy is mRNA. This temporary set of instructions will be sent to the ribosome, where the actual protein is made. Once transcription is complete, the freshly made mRNA exits the nucleus. In eukaryotic cells, transcription happens inside the nucleus. But in prokaryotic cells, like bacteria, there is no nucleus. Instead, transcription occurs directly in the cytoplasm. Despite this difference, the process works the same way in all living organisms. Now that we have our mRNA, it's time for the next step, translation.
So now that we're in translation, the ribosome acts like a factory. It's made up of 2 subunits, a large subunit and a small subunit. These work together to read the mRNA sequence and assemble a protein, 1 amino acid at a time. The ribosome reads the mRNA in segments of 3 nucleotides called codons. Each codon is like a word in a sentence, carrying a specific instruction for building the protein. But how does the ribosome know which amino acid to add? This is where transfer RNA comes in hand, or tRNA. Think of tRNA as a delivery truck. It carries amino acids to the ribosome, matching each codon with its corresponding amino acid. At the end, the tRNA has an anticodon, a sequence of 3 bases that complements the codon of the mRNA. At the other end, it carries a specific amino acid. When the correct tRNA finds its matching codon on the mRNA strand, it locks into place like a puzzle piece. The ribosome then forms a peptide bond between the amino acid and the one before it, linking it together in a growing chain. This chain will eventually fold into a functional protein. This process repeats codon by codon, amino acid by amino acid, until the ribosome reaches a stop codon. This signals that the protein is complete and is released from the ribosome to be folded and processed into its final shape. Once again, there are 3 types of RNA. mRNA, which is used to be carrying DNA to the ribosome, tRNA, used for bringing amino acids to the ribosome, and rRNA, creating the part of a ribosome itself and helping building the protein. In the case of melanism, this process results in the production of proteins that regulate pigment. The more eumelanin producing proteins there are, the darker an organism's fur, feather, or skin will appear. This is how a simple change in DNA can have a visible real-world effect.
Now let's connect everything back to melanism. We know that proteins play a crucial role in determining an organism's trait, and we've seen how the MC1R gene regulates pigment production. But what happens when this gene mutates? Normally, the MC1R gene acts like a switch, turns on and off to regulate the production of eumelanin, the pigment responsible for dark coloration. However, in some animals, a specific type of mutation called a point mutation alters just a single nucleotide in the DNA sequence. This tiny change can have big effects. This specific type of point mutation is called a missense mutation. Instead of stopping protein production entirely, it changes how the protein functions. In the case of melanism, the mutation locks the MC1R gene in the on position, continuously signaling the cell to produce more eumelanin. The result? Darker fur, feathers, or scales. But not all melanism mutations are beneficial. While melanism often provides advantages like better camouflage or improved heat absorption, it can also come with serious drawbacks. Take the melanistic barn owl, for example. This owl has a TYRP1 gene mutation that causes an increase in eumelanin production, resulting in darker feathers. But this mutation has negative consequences for the owl. For a nocturnal predator like the barn owl, lighter plumage is crucial for stealth. Darker feathers can make the owl more visible to prey, reducing its hunting efficiency. The darker coloration also makes it easier for predators to spot, increasing its risk of predation. As a result, melanistic barn owls face lower reproductive success. Studies show that they may have fewer offspring due to these survival challenges. This emphasizes that not every genetic change is a good one. But as you know, it's not all just about negative attributes. For a nocturnal predator like the black panther, darker pigmentation is crucial for being stealthy. The dark fur helps the panther blend into its dense jungle or forest habitat, making it less visible to prey. This camouflage gives the panther a distinct advantage when hunting in conditions, allowing it to be more successful in capturing prey. As a result, melanistic black panthers have a higher chance of survival and successful hunting, which translate to better reproductive success.
Mutations like melanism remind us that even the smallest changes in DNA can lead to extraordinary transformations. So remember, the next time you see a black cat, a dark feathered bird, or even a black panther, remember, it's not just luck, but it's a mutation.